Hi, my name is Jacob Elliott. I'm a PhD candidate in the graduate program in acoustics at Penn State, and today I'll be discussing my research entitled Histotripsy Bubble Dynamics in Anisotropic Hydrogels and Bovine Tendon. And to give a brief overview of this work, I'll give a brief introduction to histotripsy, which is a mechanical modality of focus ultrasound. This utilizes the creation, oscillation, and collapse of cavitation bubbles in order to liquefy tissue within the focal volume of a HIFU or high-intensity focus ultrasound transducer. While this has been proven successful in soft tissue, collagenous tissue, such as tendon, is extremely resistant to histotripsy, and this has been attributed to its high elasticity and anisotropy, as well as its low water content. This video that we're looking at here shows a bovine tendon undergoing histotripsy treatment, and in the blue focal region corresponding to the focus of the HIFU transducer, we see these bright spots indicating that bubble activity is occurring, but this is not conducive to fractionation. And it's important to note that achieving histotripsy in these tissues would demonstrate the feasibility for creation of controllable microdamage to induce a healing response in the case of a chronically injured tendon. And in turn, this would show potential for focus ultrasound as a non-invasive therapeutic for tendinopathies. So therefore, our ultimate goal here with this research is to develop a more fundamental understanding of the bubble dynamics within collagenous tissue, more specifically why this bubble expansion and collapse that we know is occurring is not resulting in the fractionation that we're looking for. And so because of this, one of our goals is to develop a transparent hydrogel that closely mimics these collagenous tissues, and namely mimics the anisotropy of these tissues, as typically that's lacking in these types of hydrogels. Commonly used gels such as polyacrylamide, which we see on the left here, as well as agar gels, are generally isotropic. So here we've also chosen to look at fibrin gels whose well-organized fibrous structure was anticipated to result in a higher degree of anisotropy. These gels were treated with histotripsy as well as bovine tendon samples, an example of which we could see here. And during histotripsy, the cavitation activity was monitored using passive cavitation imaging, or PCI, as well as high-speed photography in the case of the transparent hydrogels. To be more specific, the polyacrylamide gels were fabricated with increasing concentrations of acrylamide, which increases the stiffness of these gels. And in the case of fibrin, the water content was tuned by either adding less initial water content to its formulation or allowing these gels to dehydrate over the course of 24 to 48 hours before embedding in a polyacrylamide gel to be treated. After fabrication or dehydration, the sound speed was measured in each axial direction using 1 megahertz contact transducers, and then calculating the time difference between the peaks in the waveform. A representative example is seen here, and these correspond to the first and second reflections of the transmitted pulse in the holder of these gels with a known dimension. So once we know the sound speed, we could then back out the anisotropy, which is the ratio of elastic moduli, which works out to be the ratio of squared longitudinal sound speeds in this case. Finally, after this, the gel and tendon samples were then placed at the focus of a 1.5 megahertz HIFU transducer and treated with conventional boiling histotripsy parameters, which we could see here. With each HIFU pulse, 2 milliseconds of radio frequency or RF cavitation data was collected by a passive imaging transducer with a bandwidth of 4 to 7 megahertz. And this allowed for peak cavitation emission energy to be post-processed, compared across all of the gels and the tendon samples. And simultaneously, in the case of the transparent hydrogels, high-speed photographs were captured in order to evaluate the growth and size of the resulting bubble cloud during histotripsy treatment. The first observation we could gain from this study is that overall fibrin gels demonstrate less cavitation activity than polyacrylamide. So peak cavitation activity or peak cavitation emission energy in fibrin gels on the left here, as opposed to polyacrylamide on the right, was found to be over 35% lower on average in the fibrin gel. And shown here are representative passive cavitation images for each gel with the corresponding peak focal energies that are significantly lower in fibrin. Additionally, as we adjusted the water content of fibrin gels by either adding less water content in its formulation or allowing them to dehydrate, we see that a decrease in water content corresponds to a decrease in peak cavitation emission energy, as well as just a decrease in visual cavitation activity that we could observe in high-speed photographs. So when we compare the dehydrated fibrin on the left to standard fibrin on the far right here, we see a significantly less violent cavitation activity overall, as well as quantitatively a decrease of over 60% in the peak cavitation emission energy. Additionally, we see that the reduction of water content slightly reduces that violent cavitation activity and also reduces the peak cavitation emission energy by a little bit less than dehydration, by about 50% compared to these standard fibrin formulations. And when we take a look at the statistical analysis, we see that standard fibrin formulations, while not significantly less, do demonstrate less cavitation activity or energy than polyacrylamide. However, dehydrated fibrin, as well as fibrin formed with less initial water content, were significantly less than both gels and very similar to that of tendon. However, the degree of anisotropy, which again is calculated as the ratio of squared axial sound speeds, was unaffected by the initial water content, but was actually significantly higher in dehydrated fibrin than all other gels, and approached about 16% of what has been measured for bovine Achilles tendon. In conclusion, we can note that dehydrated fibrin gels model quite well the cavitation activity occurring in tendon during histotripsy. By adjusting the water content through either recipe changes or dehydration, we could predictably reduce our cavitation energy, and as well, the dehydration of these gels is shown to induce this anisotropy, potentially due to a shrinking of the volume between these well-oriented fibrin fibers, which could constrain the bubble expansion and therefore hinder the strength of bubble collapse. And with that, I would like to thank NIH for their support of this work, as well as my lab mates in the Basel lab at Penn State. Thank you.